Today I'm going to talk about, if we can figure out this, I'm going to do something a little bit different. We have several projects going on in the lab that range from embryonic stem cell work to work with standard genetic models. I've chosen two of the major projects in the lab and then a third that I'm also going to do. So there are really three parts to the talk today. The first part, yeah, we need a little bit better light. The first part will be on a model of microcephaly that we've been working on for about 10 years now, and I just don't talk about it enough.
Sorry,
advanced, I don't want to say, it's advanced technology here, clearly. Is that voice activated?
Okay,
Perfect. I think it's this light that makes it just difficult. So the images will be a little bit dimmer than usual, and that's unfortunate because the middle part of the talk are something like this, which maybe you can't see. I know there's an interest in astrocytes here. Can we just flick it? No, we can't flick it off just for a little bit every now and then. No, we'll do it one time in the middle, so you can appreciate. Not this time. One time in the middle, so you can appreciate this image fully. I'm not going to tell you yet how this is generated, but this is a new technology that we've developed for the developmental biology aspect of our projects. All of our projects, though, start usually with some genetic observation, genetic understanding. We try to understand the physiology or changes in physiology through developmental biology. So this first story starts actually with a fortuitous mutation, not for the rats that had the mutation, but fortuitous observation in the animal colony for the graduate student who was working on the project. And it was this phenotype. You can see it's not very subtle. He found that the brains of this animal, autosomal recessive, were about half the size of normal. And it very clearly was similar to a disorder in humans, also autosomal recessive primary microcephaly. And in humans, we know that there are 7 autosomal primary microcephaly genes, and 6 of them actually have been identified. ASPM is the most common, and WDR62 is the second most common, actually just reported in Nature Genetics and Nature just this last week and last month. So this got us started thinking about, we were always interested in neurogenesis and the control of the establishment of neuron numbers, and this phenotype told us that we had an entrance into a system that was going to tell us something very fundamental about mechanisms that establish the appropriate number of neurons, the appropriate size of the brain. So our guide is the dramatic phenotype. There's another aspect of the phenotype that's relevant, I think, to this group, and I think not taken advantage of enough, this particular model. It's in our colony and in one mutant mouse rat resource. This is a mutant rat, so there are certain advantages for electrophysiologists for that. But this rat, I even call it a mouse, this rat has severe seizures starting really early in age. So by postnatal day 9, they're having about 4 to 5 seizures an hour, lasting anywhere from 1 minute to 2 minutes. And it continues through that neonatal period, and then they die sometime around postnatal day 27. It has sort of an interesting pharmacology. Valproic acid will decrease the duration of them somewhat. We occasionally will do some screens with other AEDs, and we've found no AED that will block the seizure in these animals. So it has some similarities to the severe types of neonatal seizures that are seen with large cortical malformations or global malformations of development. They're also, during the interictal period, these classic sort of spike discharges. We don't really know what's responsible for the seizures, but we've done a lot of anatomy on this animal over the years, and there is a conspicuous absence of interneurons and a very conspicuous hypertrophy of inhibitory interneurons. Here's just the massive decrease in interneurons and then the change in the interneurons themselves, which suggests, a simplistic model that just the decrease of these interneurons might be the reason for the seizures. So the protein that's mutated, the gene that's mutated, in this animal codes for a protein called citron kinase. And again, it's a fairly understudied protein. The mutation itself is in the first exon, and it results in a nonsense premature stop codon. So it's essentially a null. There is no citron kinase in the mutant. And what we know through antibody studies is that it has a very conspicuous expression pattern, so that the protein during metaphase polarizes to the ventricular zone surface, where all of the neurons that will make the pyramidal, all the progenitors that will make pyramidal neurons in the brain are generated. It then becomes localized to both sides of the cytokinesis furrow and ends up right at the mid-body junction, where a cell will ultimately undergo cytokinesis. And at that cytokinesis furrow in mid-body, it actually co-localizes and biochemically interacts, studies I'm not showing you in detail, but biochemically interacts with ASPM, which is the primary human primary microcephaly gene. And so, in fact, all of the human primary microcephaly genes, except for one now, localize to either the centrosome during the cell cycle or the mid-body. And because of that, a model has emerged from Chris Wood's lab primarily, that primary microcephaly is fundamentally a defect in cell cycle. It's a defect in the orientation of centrosomes and perhaps the orientation of the centrosomes to the cleavage furrow, which has to occur right in the middle, or it's sometimes localized in different positions for cells to appropriately inherit the information that they need to then either go on and differentiate as neurons or go on and not differentiate and stay self-renewing progenitor cells. And we were pursuing that as a mechanism for a long time, but I want to talk to you today about some very, very new data that's done by, and we're still pursuing that cell cycle approach, but new data from Matt Girgenti, a graduate student in his third year. And in fact, all three stories I'm going to tell you today are projects from third-year graduate students that they've done really just in the last year. And there were things when he entered the lab that always didn't quite make sense for us, that citron kinase would only have a function in cell cycle. And those included that citron kinase, it's expressed really early in development, right in cytokinesis furrows, and there's no problem with cytokinesis or early cell division in the early embryo. The early embryo and the early embryonic brain are fine. Skin cells express a lot of citron kinase right in the same places. Those cells are fine. And then there was this series of papers that came out studying the Drosophila mutant. This is an example where the Drosophila mutant came out after the rat and the rodent, the mouse mutant as well. And in the Drosophila mutant, there were two interesting observations. First, there were some alleles that were not null alleles, but they were loss of function alleles with no cytokinesis defects in some tissues. And in those tissues that had cytokinesis, that did not have cytokinesis defects, gene array studies showed massive changes in epigenetic regulation of gene expression. So that decoupled cell cycle events and cell cycle regulation and regulation of gene expression. So we thought maybe, in fact, there is a connection between citron kinase and something other than cell cycle. And there was another missing piece of this whole puzzle, and that was citron kinase had no known substrates, even in cell cycle, that could regulate cell cycle. So we had a kinase without well-known substrates, and we had a hypothesis that there might be changes in the transcriptome or epigenetic regulation. We didn't know for sure. Our approach was then to do these two screens. Take embryonic day 13 forebrain, which is a time when it's impossible to phenotype the brains. They look, for all intents and purposes, every assay we've done, the same between the wild type and the mutant. And use RNA-seq whole transcriptome comparison for expression levels. Now, for those of you who aren't familiar with RNA-seq, it's really the best way to look at changes in expression level now. And if given a choice between RNA-seq and microarrays, always choose RNA-seq. The data is, I'll try to convince you, the data is unbelievably reliable, and also it's incredibly useful. It's also comprehensive in the number of transcripts that can be measured. So when we did this screen, we had sequence tags for 23,000 expressed genes in the fetal rat forebrain. And that represents 36 million data points. So you get really a full representation of pretty much every gene that's expressed. And if you do bootstrap a comparison between wild type and wild type, two different ends, the correlation between expression levels measured between two separate animals is remarkably good. And using this data with n's of 4, we found that in fact 9,000 transcripts are upregulated in the mutant prior to any phenotype onset. And about 2,000 are downregulated significantly by ANOVA with n's of 4 in about 2,000 transcripts. So this is a bit of a lesson, I think, to all of us, that we think our phenotyping by morphology or even physiology is always going to be predicting lack of any change. But in fact, here we have two tissues that we can't tell a difference between any measure. We're fairly good developmental biologists, but yet at the transcriptome level, the changes are massive. So first question is, what's responsible for those changes? And we don't know yet, but it's sent us on a search. And the other thing is we wanted to make sure this wasn't artifactual. We didn't think it was, but we used qPCR to validate 85 randomly chosen genes with different fold levels of expression. So here we're plotting the RNA-seq data fold change versus the qRT-PCR fold change. And they lay right on top of each other for the most part. So what we really essentially did is we did 23,000 northern blots. And we haven't found any examples where it's not validated by a qRT-PCR. And we continually go back to that data set to compare what transcripts might be different. We still validate by qPCR when we need to. Now, another thing you can do with this kind of data set is you can ask, of all of these genes that are changed, is there any differential representation within different functional categories that the changed transcripts are in? So by looking at gene ontology categories for cell processes, we find that in 3 categories, there's a much greater representation of changed transcript types for these functional categories. There's still plenty of changed cells, so this means that there are twofold more changed than unchanged in these functional categories, which are unlabeled. There's still plenty of changed transcripts, but in these 3 categories, so there's some specificity to this transcriptome change. These 3 categories are way overrepresented. And of course, the first thing, since this is primary microcephaly, the first thing you would ask is, what about the known primary microcephaly genes that are in human? Are any of those changed in expression? And so when the data first came in, this was about 1.5 years ago, we looked at the 5, at that time, known human primary microcephaly genes and the transcripts that they encode, and essentially found no difference. You know, there might be a little difference here, but no difference to really pay much attention to, even though things in those categories are often changed. And within this last month, this gene, WDR62, was defined as actually the second most commonly found cause of primary recessive microcephaly. And it was found in 2 separate papers by the Woods group and also a group at Yale. And when we go to our data set and look at that transcript, there's a huge decrease in the mutant. So we think we maybe, in our doing this, maybe have placed one of the other known microcephaly genes in this transcriptome pathway, which obviously leads to the question of how would a kinase that we know is important for cytokinesis in some cells, in some later progenitor cells, and some cell types, what is the substrate that would result in the absence of any cell cycle defect, such a wide number of yet specific mRNA changes in some categories? It's either going to be signaling that is maybe directly in transcription, maybe epigenetic change, maybe RNA processing or stability. And we reasoned that the key to understanding that connection is going to be to define what the substrates are and find the substrates for this kinase that really had one poorly characterized substrate. So for this, Matt used fairly new technology for finding new substrates, phosphorylation substrates. So it's a mass spec approach we had mutant neural progenitors that we can grow in cell culture and differentiate and recapitulate some of the phenotype. We can rescue the cells if we transfect in an active kinase, and we have a dead kinase that we can use as a control, nice transfection control. And we reasoned that if we take mutant neural progenitors in one population, transfect in an active kinase in cells that don't have that kinase, and in the control population put in a dead kinase, so everything is nicely balanced, and then look for the peptides essentially that are phosphorylated in this condition, the active kinase condition, they're absent from this condition. Now note, most of the phosphorylation groups are going to be on residues that other kinases are phosphorylating. There are plenty of other kinases in that cell. So there's a big population that should be the same and a smaller population that should be different. Goes through, a key step here is a titanium dioxide bead purification that serine, phosphates that serines and threonines bind to. So it enriches greatly. In fact, the data even shows us that directly, that it enriches to about 85% all peptides that will have phosphates at their serines or threonines. That's fractionated, goes through essentially a standard MS-MS protocol with first a liquid chromatography separation. Individual features and peaks are taken from that liquid chromatography, and you get both a mass abundance measure and a sequence within those features, and then you match the features to known peptide sequences in the database. And so a summary of that screen, first it showed us that it worked, that there were 2,000 peptides identified that had feature scores, mass spec ID scores that were above a decent confidence interval. Above 25 was our cutoff for those of you who do mass spec kinds of things. Of those 2,000 peptides, there were 1,200 proteins that were identified. And if we compared the things that were different in the no phosphoprotein, in fact, there were no phosphoproteins increased in the inactive kinase condition above tenfold, and the max changed peptides was twofold. So we think that's probably somewhere in the noise. But in the active kinase transfected cells, there were 8 phosphoproteins that were greater than tenfold enriched. And those greater than tenfold enriched, there was one of them, and they range from 32 to 9.5, so I rounded up on this one. So there was one that was 29-fold enriched, and in fact we had 3, and it was the only one that had 3 separate peptides that were represented in the mass spec. And I think you can see, we're working on this now, that there seems to be a motif in here as well. There's this serine and proline, serine and proline, serine and proline in several positions. So we're working on that. We actually, to really define the full motif, need more than the peptides we have here. So we're doing other approaches than that. But it looks like it's a serine, proline, something motif for this kinase. What does this kinase do? Well, it turns out that this isn't a kinase. What does this protein do? It's a nuclear repetitive, serine-arginine repetitive matrix protein, and was first described by Phil Sharp, and he described it as a co-activator of pre-mRNA splicing. Not affecting all splicing of mRNAs, but just some. And it's still actually being worked out which RNAs it's important for splicing. Here's another advantage then of the RNA-seq data set. So because the RNA-seq data set is a read of sequence tags to individual exons, you can go to that same data set and ask, are there any splice variants within that data set? We didn't have reason for doing this initially, but now that we pulled down SRRM1, we have reason for looking at it. So our approach to this informatically was to count the tags in each exon for every gene that was differentially expressed. We could do it for all genes. We cut down our search a little bit. So we just looked at the differentially expressed genes and counted up all of the individual sequence reads for each exon. And it's only in exons because we did this on mRNA. So we did all the sequence reads for mRNA. We normalized that, and we simply asked for any gene that was differentially expressed, were there more than expected or fewer than expected reads for any exon. And there was one. There were some that were a little bit different. There was one that was an absolute difference. and that's one of the histones. And it's a histone variant I'll talk a little bit about after, but it's a histone variant that's gotten a lot of attention recently in stem cell biology, actually. It's histone H2A-FZ, sometimes it's called H2A.Z. And if you look at the number of reads in each exon for either the wild type or the mutant here is flathead, first of all, you can see that the number of reads in each exon for the flathead, and these are all the individual ends, are really very high. It's an upregulated gene. It happens to be one that's fourfold upregulated anyway. So it's fourfold upregulated relative to the wild type reads. These are arbitrary numbers, but these actually represent thousands of individual counts for each of these tags. But here in exon 4, there were only 32 reads for the mutant. So it's essentially missing transcripts that contain exon 4, whereas they're fairly evenly distributed for the wild type, which has the known 1, 2, 3, 4 exon structure. That's not surprising. But this missing exon here makes, and now that's been confirmed with qPCR, but makes a fairly strong prediction of what would be going on. A splice of H2FZ that would have 1, 2, 3 to 5 splice presents a frame shift so that there's a different C-terminus. And that different C-terminus is lacking, importantly, lysines that are normally ubiquinated, monoubiquinated, one of the 3 lysines. And when that H2AZ is monoubiquinated at its C-terminal tail, it's associated with transcriptionally repressed genes. Remember, we have 9,000 genes that are overexpressed. So most of the transcripts are overexpressed, and we're missing a splice isoform that would be predicted to be contained within nucleosomes that are under transcriptional repression. The most recent data, however, on H2AZ says it's a really very complicated histone variant, that when it gets added to the nucleosome, depending upon the nucleosome context, what other nucleosomes are associated with the complex, it can either be a very stable nucleosome or a less stable nucleosome. So it could be possible that some genes would be upregulated in expression. And what Matt is doing right now is exactly what you'd predict. We need to make antibodies to just this C-terminal tail and begin to do chip experiments to determine whether or not H2AZ is the full answer for the upregulated and downregulated genes. But it's conspicuous and it fits the bill for this kind of large-scale change in the transcriptome. So our current working model, which is something new for microcephaly, primary microcephaly genes, is that citron kinase phosphorylates SRM1, and there are clearly experiments he needs to do to pass his thesis, and then I probably won't talk about it once he does his thesis. We'll be on to something else. But he needs to show this phosphorylation, although the mass spec data is pretty strong, even to the point of getting a motif. Then we get a change in a splice isoform for a histone variant that is critical to epigenetic changes. That would change the structure of the nucleosomal content, and then massive changes in gene expression, and potentially WDR62 as well. Well, we know that's changed too. And then maybe the phenotype. Although I think the phenotype is more than WDR62. And then there's an epigenetic state that's conducive, in fact, necessary for self-renewal. Now, another thing we're doing right now is asking the degree to which other primary microcephaly genes, if we could decouple their role in cell cycle, to what degree do they interact with citron kinase or interact with a signaling process that's either coordinate with or independent of regulation of cell cycle? And just intuitively, it makes sense that a cell's cell cycle needs to be coordinated to its transcriptome state, particularly cells that are undergoing a terminal division and are going to undergo a massive transformation in cell type within hours from exiting the cell cycle. Yes?
So the experiments where you looked at the splice isoform switch, were those done on the flathead?
Yes, they're done. So they're done in both. They're done in the citron kinase null and the citron kinase containing cells.
Oh, so you did them also in those?
That's how we identified it. Yep.
Okay.
Yep. So that's a good question, though, because in the wild type Right now, we find no evidence, at least the way we're preparing our cells in our tissue, we find no evidence of the strange splice isoform, the 1, 2, 3 to 5. Okay, so that does raise the question of what is the function of this endogenous 1, 2, 3? Does it ever exist even? And if it does exist, maybe it only exists, it's not in the database. So it might only exist during very brief windows in time, during a cell's differentiation, and you would miss that. But now we have the tools to begin to look at exactly that. Does it happen in one stage of cells differentiation? That's our hypothesis. And it just hasn't been seen before. So that will help actually in the in vivo sort of switch here, whether or not this short form ever exists and changing the epigenome in a way that actually is not very beneficial. Okay, I want to switch now to the technology. And that is the technology that we've used now for a new technology for tracking cell lineage in developing neocortex. And we've expanded it to being able to label clonally related cells. And so if you didn't know before, the standard lineage model for neocortical cell generation is neuroepithelial cells, or sometimes called radioglial cells, they're kind of interchangeable now, start giving rise first to neurons, then they give rise to astrocyte progenitors that differentiate as astrocytes. That's a period that's somewhat overlapping. Then they ultimately give rise to neuroblasts that'll make olfactory bulb interneurons, and they give rise to a progenitor that'll make oligodendrocytes. And to date, the only methods for tracking this kind of lineage are either retroviruses that would integrate. Retroviruses have the disadvantage that it's hard to control the promoter in a retrovirus, and they'll express in any progenitor type, or using Cre recombinase-based lineage labeling. And the disadvantage of Cre recombinase-based lineage labeling is that you label all of the cells, not a subpopulation of the cells that you could do clonal analysis with. And it's always good to have another technology to label a lineage. And we sort of started from this idea that in in utero electroporation, which we'd been using to study migration, when you could transfect cells at the same surface where those neural progenitors exist, wait in 1 day, they migrate 10 days, even longer, they only become neurons. So for mechanisms we don't quite understand, either dilution of the plasmids or inactivation of the plasmids, which we suspect is a more likely cause, active inactivation of the plasmids. The only cells that are labeled are the first cell, what we call the immediate progeny, the first cell in that lineage, and none of the future cells, either astrocytes or olfactory bulb interneurons, are labeled. And so to get around that, Fui Chen in the lab, another graduate student just finishing his second year into his third year, Fui started to work on this kind of system using a piggyback transposase. Again, it's a plasmid-based system. It's a binary system so that there's one plasmid that codes for a piggyback transposase, and that protein then mediates the insertion of any transgene as long as it has piggyback transposase terminal repeats flanking that transgene. And it inserts it, they're not here, into TTAA sites in the genome. But there are plenty of TTAA sites in the genome. So it's essentially randomly integrating someplace in the genome, maybe slightly enriched for 5' parts of genes, but not much. And so that would give a way to integrate the plasmid into the host cell's genome, and that should avoid plasmid inactivation that we think is occurring to block lineage labeling. If that's the case, then this system should show us the same lineage. It still starts with plasmids, and it does label the whole lineage. This is one where it would be nice maybe turn off the lights, but that's okay. I don't want to mess up the video. I think you can see here, oh yeah, beautiful. I think you can see here now that we have, so we did it in two ways. So we added RFP, red fluorescent protein, along with this piggyback transposase system. So the red fluorescent protein stays in the immediate progeny, stays episomal, doesn't go into the whole, we think episomal, doesn't go into the whole lineage. And I think you can see plenty of astrocytes now that are labeled with the green piggyback mediated transposition transgene, and this is just the RFP. There are no astrocytes labeled RFP. The olfactory bulb now becomes beautifully labeled, the interneurons, so we have that lineage. Oligodendrocytes in white matter are now labeled, where we never had them labeled before. In fact, all the components of the lineage that we were missing with the straight plasmid transfection are now labeled with the integrating system. Okay, so that was nice. That can be used for, leave it off because I have one other, okay, you can turn it on and off, that's right. I've got one other slide. Actually, yeah, sure. Am I totally out of your video now in the dark? I am? Okay, turn it on for them. They work so hard to, and I've been trying to stand in one place. They told me I had to stand in one place throughout the talk. Can't tell you how hard that's been. So it would also be nice to have clonal relationships. And that's one thing we're missing with the in utero electroporation lineage approach, which is essentially an approach that would be similar to Cre-mediated recombination, only with different technology. But we thought, could we come up with a way to label these component clones and identify them unambiguously as being clonally related to each other? That has some potential functional significance because it's been shown, at least for neurons, there are increased chances for synaptic pairing between neocortically, clonally related neocortical neurons within the same column. Fascinating result, hard experiment to do. This experiment, if we could label clonal cells, in fact, we can't do neurons because we can only get the immediate progeny, but if we could label clonal cells of these other lineages, the similar kind of question could be asked. Are there functional relationships in normal physiology or in pathophysiology between clonally related cells? Okay, so our approach and our logic behind this approach was the following. As instead of adding one donor plasmid, let's add 3 donor plasmids. And with the assumption that piggyback transposase isn't 100% efficient, and it isn't, you can show it in cell culture. And so what would happen, you would have 7 possible transgenic combinations if it was just one plasmid that could go in. We should have all 3 colors, 2 color, any of 3 combination 2, or any of individual color. And in fact, it's more combinations if it's more than 1 copy that could go in. Because if you could have 2 greens and a red, that would be a different shade than 1 green and a red. So here is the result, just gotten this sum. Oh, here's conceptually simple enough. So now you should have color-coded clones because they're unique genetically integrated sites. And so there are the color-coded clones of astrocytes. I think you can see there's a little cluster of blue, of the light blue. I don't know what, we don't know what colors they got. They probably got some cyan and maybe some GFP as well. And there are some more pure clones in here that are blue. You can see some yellow clones and some green clones. If we look at one of these individually, we can see that whenever you find a pair, in fact, catching a cell that might be caught in cell division, they're always the same color. So any pair like this that's a mirror image of itself is always the same color. That helps to validate it. We've done some other clonal analyses. Here are some examples of those pairs that are of the same color. You can also see the clonal boundaries and relationships between clones because now we can see a red cluster, here's a slightly yellow and green. We would be conservative because there are a lot of green pairs. We don't really know if this is one all-inclusive clone in this, but it's either 1 or 2. What we've done, and I'm not going to show you the data, but what Fui has done is looked at really low transfected brains and looked at monocolor groupings of cells where there aren't cells around, like in these examples, in fact, to get the measure of what a real single clone is. There's one other thing I want to show you, and that's sort of a new, I think, arrangement of astrocytes that we've already discovered with this. We just developed this summer. If you look in upper layers of neocortex, you begin to see radial columns of astrocytes. So that astrocyte clones begin to arrange like we think neuronal groups do. And those astrocyte columnar clones look like they're actually made up of several clones, but each component has a radial arrangement to it. And there are some obvious functional questions to begin to ask when you have this kind of arrangement. do these clonal boundaries correspond to any kind of functional boundaries, either calcium waves that might be moving through this tissue or gap junctions connections between these, or as I was talking to some today, you could use this methodology to ask whether there's clonal expansion of individual clones after specific injuries, stroke or epilepsy. Is there any localization to clonal expansion? Because you could also quantify whether or not a yellow clone like this has now become a much enlarged yellow clone in the tissue. We can turn the light back on. Okay, so before I move on, let's keep it. Are there any questions? We can turn. Are there any questions about that technology? Pretty straightforward. Another, yeah.
Not a question, but a comment. I mean, Christian Guillaume has shown in with his viscous stimulation that in the sensomotor cortex there is obviously a restriction of the calcium waves between astrocytes within a given column.
In the barrels, yeah. So in the barrel cortex. No, this is outside of barrel cortex, but maybe could be part of that. In the barrel cortex, where there are clear differences in cell density, that's known that there are restrictions between astrocytes. We're seeing this also in visual cortex, also in frontal cortex, wherever we look, not just somatosensory cortex. I would bet these are within barrel cortex too, that are also forming that. The barrel cortex boundaries tend to be strongest in layer 4. I haven't looked in where their boundaries are, and these tend to be in layer 2 and layer 2-3. But they could be related. I think they go beyond those barrel restrictions. Yes?
Are you sure about when you call it columnar arrangement, that this corresponds to the well-known functional cortical columns? Because, of course, for example, glial columns, they might emerge simply from the dendritic bundling of, of pyramidal cell dendrites, and this might not yet correspond to the functional columns. What's your idea about?
Yeah, first, I think they're too small for true functional columns because they're not 500 microns. So if anything, they would be maybe mini columns, which in rat is difficult to define a mini column. Yeah, I don't mean by calling them at this point a columnar organization to yet associate it with the neocortical columnar organization. And it could be something like bundling to dendrites. It could also be simply a process, at least in its formation, still could be important, that the radioglia, as they transition from a radioglia into an astrocyte, are dropping off progeny as they move to the cortical surface. And so that would be a radial arrangement. It's interesting that the deep layer astrocytes don't form that same arrangement. But yeah, I agree. We don't know if they're functional in any way. They could be totally unrelated to function.
Yeah.
But you can't ask the question until you can see them. And so now we can see them. Yeah.
So I just would like to know, so did you do similar experiments into the hippocampal formation, and do you see a similar organization?
Yeah, we did do hippocampus. In hippocampus, we didn't see these radial organizations. We do see astrocytes, a lot fewer, and there are astrocyte clones that are unicolor clones, and we've seen those in dentate and in CA1. So we can confirm the technology with astrocytes in dentate, in hippocampus. We don't know why we don't get as many astrocytes. And it's not totally known whether the astrocytes in hippocampus are generated from the ventricular wall the same way that the astrocytes from neocortex are generated, a large proportion of them. But we get far more neurons than astrocytes in hippocampus with the piggyback system. Still get enough to study, but not that's kind of massive labeling. Yes?
So you're sort of alluding to this functional significance of these columns. And I mean, one thing that comes to mind is gap junctional coupling, but as far as I remember, there's no directionality in the sense of columns in gap junctional coupling, or am I in error? Or is there such a directionality in that cortex, let's say?
Yeah, none reported, and I don't think it's an absolute barrier if there's going to be one there. Generally, when you fill a cell in the middle of neocortex, you fill what looks like a circle.
Yeah,
Okay, so that would predict that coupling, and we've started to do the experiments, and we've seen the coupling boundaries go beyond the clonal boundaries. But that doesn't mean when we look at more, you know, we now have boundaries that you can look at that you couldn't see before. So maybe it peters out at the end. You know, maybe it's the margin of that coupled group that shows it. Okay, so now in the next 10 minutes, I'm going to talk about another approach another project in the lab that Alicia Shea has been working on. And this project also was inspired first by genetics and then by phenotyping, which ends up being mostly physiology phenotyping for us at this point. And I got involved in this type of project about 15 years ago now, crazy. And it was this interest that ultimately there must be an underlying cellular neurophysiology to dyslexia. It's just such a clear behavioral learning deficit that triggers in a very specific way, a specific time in development when we start to learn to try to do something that we're not hardwired to do. Right? Learning to read is one of the more difficult things we do. It's an add-on. A lot of kids fail. In fact, 5 to 17% fail at initially learning to read and fall behind in reading level fairly rapidly. And it was known through early twin studies that there is a partial genetic component and a partial environmental component. And we reasoned that once the genetic associates with dyslexia become apparent, that studying the neurobiology of those cells are going to give us some interesting insights into a very specific aspect of cellular physiology or developmental neurobiology. We didn't know which at the time. And of course, the joke is that every animal model is a model of dyslexia. There's no rodent yet that's learned to read, even in any transgenics that we've done. But they certainly have the same neurons that we use to read. And so understanding what would how their alterations in those genes would affect either the development or the physiology of those neurons is worth looking at. There are 3 genes now. There are multiple others in the literature. There are 3, DC-DC2, KO319, DYX1C1, that have SNP variants that have plausibly associated with dyslexia in at least 2 replicated studies, and for one of these genes, 4 replicated studies in different populations. And so we chose to look at these in some detail. I'm going to tell you about some recent data just on this gene, DC-DC2. I'll skip this. So here are some of the human genetic association studies, no genome-wide association study yet, that's associated DC-DC2 and dyslexia. And I think it's notable that at least 3 of them come from this university. What are the chances? I don't know. And the first report actually was out of a group from Yale, someone who got into it because they were interested in this chromosomal region. And he was the first to show the association. And then that was quickly followed up by a paper from here, again with Juha Kier, too, who's not here, and a couple other papers. There's one paper that shows not a very good association. And there's one paper here that doesn't find the same association with one of the compound short tandem repeats that this paper found an association with. But this paper still found the same associations with the single nucleotide polymorphisms that the other papers found. So this is, to us, an example of, at least while it's not perfect, we've got an Australian population, an American population, and 2 different German populations with statistically strong associations. That's about as good as a genetic association study gets. And our interest in pursuing this gene first was actually because of its structural similarity to another gene that we had been working on, which when mutated causes double cortex syndrome and lissencephaly in males, X-linked lissencephaly. And this gene, DC-DC2, is in the DCX family. In fact, it has 2 double cortin domains in the same position that DCX has. It also binds to microtubules in the same way that DCX does. And it had some evidence for being involved in migration. We did an RNAi study showing it was involved in, it could be involved in migration. And so that was the reason that we chose this gene to do a standard reverse genetic approach first. And so we made a conditional, or we had made for us a condition and have been working with it, a conditional knockout of this gene DCDC2. We targeted standard types of ways, won't go into the details. We've confirmed that it's actually a null when you take out exon 2. You get a nonsense premature stop codon. And one thing was a little disappointing, but then ultimately we think this is positive news. There were no neocortical lamination defects in this mutant. So the mouse knockouts don't confirm DCDC2 as having any role in neuronal migration. Lamination is fine. We've got lots of other assays I'm not showing here. This is just a section artifact. There actually is an intact dentate gyrus in these brains. And we thought, does this mean that maybe our RNAi studies are wrong, or maybe there's a species difference? We've gone and done the RNAi studies again that showed a migration difference in rat. We've done the RNAi studies in mouse, and we can't find a migration difference in the mouse when we do the RNAi. So there's a species difference for targeting genes between mice and rats, and this always surprises people. Aren't they exactly just the same? They're absolutely not the same. They're separated in evolution by 95 million years. Their genomic structures are completely uncomparable. Their protein sequences are the same, but they are very, very different, obviously. In fact, rats are predators for mice. So we then went ahead, since we didn't have any morphological defects, that was sort of good news, because then we could proceed with neurophysiology and some behavioral experiments without worrying about some sort of non-specific effects of gross malformations, which actually is a better model for what you might imagine in a model for dyslexia. So the one maze that did show a difference is the Hebb-Williams maze. And it's your standard mouse cartoon maze that animals learn. They first learn to do many different mazes, and then they come back and they're tested for 6 trials for all these different mazes. There are different configurations. Some are easy. This is an easy maze. Some are hard. This is a hard maze. And you count how long it takes them to learn it. Actually, it's relearning it. And how many errors do they make. And they make the same number of errors, the different genotypes, not significantly different. But the animals that are either homozygous for the mutant allele, missing DCDC2, or the heterozygous animals, they don't get any faster at running the maze over trials. They get better, they make fewer errors. They don't have motor problems because they do the Morris maze. What I like about this mutant is how many things it does well. It does Morris water maze just fine. It does object recognition tasks just fine, no difference. Tests of motor function, open field types of things. It does all those things normally, but it learns to make fewer errors. It just does it more carefully. It gets to each mistake point, and it's as if it sits there a little bit and, thinks about whether this is the right thing to do or not, and then moves on to the next thing, but doesn't actually make the error. So that begins to make you think maybe it's a speed of processing or something like that. Anyway, I'm not a behavioral neuroscientist, so my former graduate student who actually ran those experiments, Lisa Gable, has her own lab, and she's doing more experiments on those animals, and she'll figure it out for us. The expression of DCDC2, though, this is out of the GenSat back reporter library. The expression is really highest, besides olfactory bulb and a little bit in cerebellum and something down here in the pons. It's really in layer 3 pyramidal neurons and some layer 5 pyramidal neurons. And so we also have done an electrophysiological assessment in these animals, and we've targeted layer 2-3 cells in somatosensory cortex. For really no other reason than that's where the expression was highest. And so it could serve as a generally decent model for asking what's going on in cells that now are lacking DCDC2. And this started out as Alicia's neurophysiology training project. And after about a year of recording, it's turned out to be a clear a clear project. There is a significant shift, first of all, in resting membrane potential, which is pretty obvious, and a significant change in, which I'll get to in a second, a significant change in action potential firing frequency that's independent of the change in resting membrane potential. So here they are with adjusted membrane potentials. In fact, these are two cells that happen to have the same resting membrane potential, so there's no extra current being injected. And what happens in the knockout cells is they just come to threshold with less current. There's a slight input resistance change in these cells too, so that could be part of it. But it's more than that because they attain a higher firing frequency with maximum current injection than the wild-type cells. They seem to be more excitable by this measure, can fire much higher frequency action potentials. And that was interesting difference, but it doesn't really quite fit. It fits with a model for maybe epilepsy, but not so much with dyslexia. So we began to ask, what might be a connection between temporal processing and change in firing rates? And so we began to look at trial-to-trial reliability or precision in when the action potential is fired. And here are some examples. Here's a wild type. And you can see when you overlay where the action potentials are occurring in 300 picoamp current pulse, they're generally at the same time. In a knockout cell, they tend to be jittery from trial to trial, sometimes happening a little bit before, sometimes a little bit later. So here's a typical wild-type cell. They all behave like this in our hands in these layer 3 cells. At each trial, this is 10 separate trials, they all fire their action potentials at about the same point in time from trial to trial. In the knockout cells, we see two types. One is a lagging type, which gets a little bit slower, and that was about 30% of the cells. And another one is a more variable type, which might fire a little bit faster, might fire a little bit slower on the next trial, but there's more variability on when that action potential will occur for the second, third, fourth, fifth action potential. So the way Alicia quantified this was to just take the coefficient of variation for when the action potentials occur across this time. And this is just an example of when you increase the stimulation, you still see these changes. The wild-type cells now, even though they're firing at a much higher frequency, they still remain regular. The knockout cells, even though they're firing at a high frequency, now they still either have the lagging or the slightly randomized effect. And if you plot this as a function of coefficient of when the action potential is occurring, actually which action potential it was, so which action potential in 50 to 500 milliseconds, and the coefficient of variation across, I think these are now 20 trials, you see this huge increase in the variation, trial-to-trial variability of when the action potential occurs during a train, even at the first pulse. That coefficient of variation is actually decoupled from resting membrane potential. So any change in resting membrane potential doesn't affect this. And it, in fact, is there for all current injection amounts, even when the action potential, so in this case, in the knockout, at 300 picoamps, it's firing about the same number of action potentials as the wild type or the reverse. The wild type at 500 picoamps is firing, in fact, more action potentials than the knockout at 300 picoamps, but the coefficient of variation is still much higher when they're firing the same number of action potentials. So even balanced for action potential number, there's still a massive change in coefficient of variation. So that's gotten us thinking, and now we're doing in vivo experiments, that DC-DC2 maybe is involved in the temporal precision at which action potentials fire from trial to trial. And obviously, that would degrade in a temporal way the fidelity of any signal coming in. So we have questions of how, in the future questions, Alicia will ask for her thesis, is how does DC-DC2 change relatively fundamental neurophysiological property? Could be trafficking of channels, either with a K, C, or with a K or not with a K. It could alter channel modulation through some cytoskeletal change. We know it associates. Or maybe it could be a morphological change, and what we're dealing with is changes in cable properties. We could look at that one. We're still working on the other ones. And we can find no changes in the pyramidal neuron structures primary, secondary, tertiary dendrites. So we don't think it has anything to do with a morphological change in these neurons. We have used a whole transcriptome analysis, and just because it is, at some point, it's the ultimate phenotyping. Yeah, it doesn't really tell you much. The important things are that even in this single gene knockout, there are 530 upregulated and 492 downregulated genes. Okay. We don't know if they're primary or secondary. There's an activity difference here, so maybe we shouldn't be surprised. And most of them are the biggest upregulators are 5-fold, and the downregulators by 5- or 6-fold. It is good to see that it's not as much as the citron kinase mutant, where we can't even phenotype it. But at least this mutant does create some changes. There is maybe a clue as to what's going on. If we look at the functional categories, this time based on molecular categories, these aren't individual genes, the top 10 of 25, in the top 25 upregulated categories are in these ion channel, neurotransmitter receptor, transporter gene families. And we don't think there's a direct effect on transcription, could be, but we think this is probably more likely a compensatory effect of the change in physiology, some sort of homeostatic change. We can follow up on that once we connect all the dots. Okay. So that leaves us with a summary of this part. What we have is this DC-DC2 mutation, which is the gene that right now has got the best evidence for being associated with dyslexia. We might have altered ion channel expressions, probably indirect. We've got some increased neuronal excitability and degraded temporal precision, and now we think that would lead to altered processing and maybe altered learning strategies. Clearly, the experiments that need to be done next are not just from these mechanistic sides, try to figure out which channels are responsible for this shift in temporal precision. And there are some candidates out there. If you block with 4AP some channels, you'll get shifts in temporal precision. And so we have some candidates to look for there, but also this question of what are the in vivo consequences of sensory processing. Again, we think they'll be subtle and not absolute, but it might give us clues as to what the dyslexic brain, how it might be processing stimuli either for learning or sensory processing a little bit differently. And we predict that it would be an alteration in temporal precision as opposed to an alteration in overall excitability. Okay, so the people who really did the work I talked about today are Matt Genti, Fui, and Alicia, and these are some other people in the lab. And I'll finish there for questions.